Thank you for joining me here at Rare Disease Week. My name is Jenny Ekholm, and I'm part of the PacBio team. I will give you an introduction into HiFi sequencing and present on how it can help to increase understanding of rare and Mendelian diseases. There are many types of variants in the genome, ranging from single nucleotide variants, deletions, insertions, tandem duplications, interspersed duplications, inversions, translocations, and copy number variants. These are all very important because they are what makes us different from one another and also what contributes to both health and disease. And even though single nucleotide variants are by far the most common variation, due to their size, they still only make up about 5 megabases of the human genome. Aside from CNVs, 3 megabases of the human genome is made up from indels, and due to the larger size of structure variants, they actually make up 10 megabases of the genome. In order to detect the smaller variants like CNVs and indels, high accuracy is needed, and therefore these have traditionally been detected using short-read sequencing. For larger structure variants, where read length is key, long-read sequencing has been applied. However, with HiFi reads, you no longer need to choose between read accuracy and read length. You can now get highly accurate long reads that can discover all variant types, ranging from CNVs to large structure variants. So how does one generate HiFi reads? In order to generate highly accurate HiFi reads, every molecule needs to be sequenced at high coverage. The sequencing polymerase has strand displacing capability and will therefore open the SMART bell into a circle, allowing for the sequencing of both forward and reverse strands. The sequencing reaction continues until the sequencing run has completed. The sequencing errors, here indicated as red dots, are random, and therefore they will be washed out by increasing the number of passes around the SMART bell. The consensus accuracy for a HiFi read with 10 passes is 99.9%. Not only can HiFi reads capture all different types of variants, but they can also detect variants that are often missed with short reads. Due to the read lengths, short reads usually misses over 50% of the structure variants, particularly long insertions and variants in the repetitive regions of the genome. Here is an example of a structure variant. On the top, you see PacBio HiFi reads, and on the bottom, short reads for the same sample. HiFi reads are long enough to span the entire region, and therefore the 1700 base pair deletion can clearly be detected, whereas the short reads are not mapping well to the region, and this is because of an underlying repeat expansion. In addition, HiFi reads enable phasing of the alleles, confirming that this is a homozygous deletion. Even though short reads are commonly used for CNV and indel detection, they still miss a portion of them, especially the ones that are located in difficult to map regions of the genome. Here is an example where the CNVs are missed by short reads. The STRC gene, which is known to cause congenital deafness, can easily be sequenced and the alleles phased using HiFi reads, shown here in the middle. However, on the top, Illumina short reads have gaps, and on the bottom, ONT long reads have many errors. The reason for the difficulty in this region is that part of the gene, here indicated with arrows and dashed lines, has a pseudogene copy that is very similar in base content, causing mapping errors between the pseudogene and the functional copy. In a recent publication in Nature Reviewed Genetics by Logson and colleagues, they concluded that short reads missed 47% of the deletions and almost 78% of the insertions. And this was after applying 11 different variant colors specifically designed to detect these variant types in short read data. Overall, it has been shown that NGS short reads miss 5% of the medical exome. This might not sound like a big number, but it does affect 193 medically relevant genes. However, with the higher precision and recall, HiFi reads are able to resolve most of these regions. Because this is a single molecule sequencing technology, the sequencing data will have a read length distribution, with most reads being around 15 to 16 kb for HiFi sequencing run. The median accuracy of the data is over 99.9%. This means that if you have one read that is 19,820 base pairs long, then you will only have about 8 errors in the read, indicated here in red. Therefore, this read is highly accurate with a quality value of 34. The complete and accurate HiFi data type that is generated by the Sequel IIe system can be applied across many different applications such as de novo genome assembly, variant detection, targeted sequencing, RNA sequencing, and looking at complex populations. These applications can be used across many different types of samples and also across species. One of the areas that have benefited from HiFi sequencing is rare and Mendelian diseases. Here you can see a historical representation on explanation rates across technology evolution that is used for mapping rare diseases. The higher the genome resolution, the more comprehensively they can detect variants in the genome. With chromosomal karyotyping, 5% of the rare disease cases were explained. With microarray technology, that number increased to 12%. With short-read sequencing, that number is around 30% for exome sequencing and below 50% for whole genome sequencing. For HiFi sequencing, that number is around 67%, based on what we have seen from collaborations, presentations, and publications to date. This number could increase further in the future, since the analytical tools and databases, especially for structured variants, will continue to improve over the next few years. As I mentioned, numerous groups have applied this approach to rare and Mendelian disease successfully to help map disease-causing variants where they have previously gone missed with other technologies. In 2017, PacBio whole genome sequencing was used for the first time to identify a genetic cause for a Mendelian disease. The study case was a 22-year-old male with recurring heart tumors, and despite applying several genetic screening methods using NGS, no disease locus was identified. You and Ashley from Stanford University applied PacBio long-read sequencing and found a 2.2 kilobase heterozygous deletion in the PRKAR1A gene that has been linked to autosomal dominant carnic complex disorder, a disease matching the study subject's symptoms. You and Ashley commented that PacBio long reads allow to illuminate dark corners of the genome like never before. Another example of this is a recent study by Jeremy Smuts and Greg Cooper from Hudson Alpha. They described the NGS unexplained study subject of a pediatric female with intellectual disability, seizures, and speech delay. In previous studies, no causative variant had been identified. However, by applying HiFi sequencing, they found a 7 kb insertion along with the precise breakpoints as the underlying genetic cause. In another publication by Dr. Matsumoto from Yokohama City University, described a now 12-year-old girl who was diagnosed with intellectual disability at 5 months old. HiFi sequencing helped Dr. Matsumoto to identify a copy-neutral 12 kb inversion as the causative genetic variant. Also, because HiFi reads are so accurate, it enabled the researchers to determine that the inversion was a de novo event, the precise breakpoints, and the mode of inheritance. And in this case, the variant was inherited from allele 1 from the mother. In addition to these studies, there are some large consortia initiatives that have implemented PacBio long-read sequencing to their rare disease research studies. For example, SolveRD, funded by the European Union, is committed to sequencing 500 individuals using HiFi sequencing. In the US, there are two large consortia applying HiFi sequencing to their studies. Children's Mercy Kansas City are planning to sequence 100,000 individuals total as part of the Genomic Answers for Kids project. And Discovery Life Sciences at Hudson Alpha Institute for Biotechnology has started to apply long-read sequencing to thousands of human genomes to characterize both common and disease-causing variants. So in summary, I had given you a few examples on how PacBio HiFi sequencing has been used by the scientific rare disease community. HiFi Reads introduces a new paradigm by enabling the development of rare disease diagnostics, where the researchers can now access all types of variants found in the human genome. This means now that one genetic assay can be used to unfold genetic mysteries that previously have gone unexplained. To learn more about the detailed rare disease workflows, I invite you to watch Christine Lambert's presentation on the wet lab workflow, as well as William Borwell's presentation on the data analysis workflow. And if you're interested in learning more about how to start your own project using HiFi sequencing, then you can choose to visit us on our webpage on rare disease, variant detection, or HiFi sequencing. Alternatively, you could also reach out to us and speak to a scientist about your project by visiting pacb.com/scientist. Thank you so much for your time.